And now we move on to the thylakoid membrane, which function specifically to conduct light absorb, transferring the light absorbed or the light energy into the chemical energy. We have gone through the definitions of photosynthesis, the component involved in photosynthesis, what are the elements required for photosynthesis. There are two specific or individual processes involved in photosynthesis. And here we come to the light reaction process, which mainly associated with the thylakoid membrane. Because in the early slide, I did ask a question: what is the specific structure which conduct photosynthesis? Is it the leaf? Is it the cell? Is it the chloroplast? Is it the thylakoid? Is it the granule? So the answer is the thylakoid membrane. Thylakoid membranes contain many photosystems, consisting of a cluster of chlorophyll and accessory pigments molecules surrounded by various proteins. Thylakoid membranes contain many photosystems, consisting of a cluster of chlorophyll and accessory pigment molecule surrounded by various proteins. There are two photosystems working together during light reaction, namely photosystem 2 or PS2 and photosystem 1 or PS1. Each type of this photosystem, they has different electron transport chain or ETC, which located adjacent to it. And each of these electron transport chain, they consist a series of electron carrier molecule which embedded in the thylakoid membrane. Within the thylakoid membrane, the overall path of electron is following the followed pathway from photosystem 2. They are following electron transport chains 2, next to photosystem 1, following the electron transport change 1 to produce NADP. Therefore, we need to know in light reaction process, specifically on the thylakoid membrane, they consist a cluster of chlorophyll as well as accessory pigment molecule which work specifically in two photosystems. The photosystems are photosystem 1 and photosystem 2. And this photosystem, they has different electron transport chain and consists a series of electron carrier molecule. This is how the thylakoid membrane embedded with photosystem 2 electron transport chains 2, photosystem 1 and electron transport chain 1 looks like. The composition of the system involving, once again, photosystem 2, electron transport chain 2, photosystem 1, electron transport chain 1, together with the light energy to be absorbed, water, oxygen, carbon dioxide, ATP, and ADPH, all are being conducted in the thylakoid membrane, thylakoid compartment, as well as in the stroma. Let's move on into the detail of light reaction process which associated with the thylakoid membrane. First of all, light energy which absorbed by the chloroplast, they will eject electron from photosystem 2. Now we know the purpose of absorbing light is not only to provide the energy packets, which means the light which the chloroplast absorb, they come in the form of packets of energy called photon. So the purpose of this absorption is to eject electron from photosystem 2. The ejected electron from photosystem 2, they will hop or jump from one pigment to another pigment until they reach into the reaction center. I repeat again, the energy is in the form of electron which ejected by the absorbed light on the thylakoid membrane. Once ejected from the photosystem 2, this electron, they will hop or jump from one pigment to another pigment until they are funneled or channeled into the reaction center. Once the electron or this energy reach the reaction center, the energy from light will boost an electron from one of the reaction center of chlorophyll to the primary electron acceptor, which captures the energized electron. It means that the hopping electron or this energy, they need to funnel into the reaction center. Why they need to be funneled into the reaction center? Because the energy will be excited or kept or stored in this reaction center. Let's repeat again. One photon or packets of energy being absorbed by the chloroplast, especially on the thylakoid membrane. Once they strike on the thylakoid membrane, the electron will ejected from photosystem 2. And this electron ejected from photosystem 2, they will hop from one pigment to another pigment until they has funnel or channel into the reaction center. Based on this process, this electron, they will lose the energy because they are indirectly transferring the energy while they are hopping from one pigment to another until they reach to the reaction center, which means levels of energy or amount of energy may be reduced. Therefore, once they are in the reaction center, this energy will be excited again to be kept for other metabolism process. When the electron reach into the reaction center, this electron will be excited in order to keep the energy in the reaction center for the purpose of other metabolism to take place. The fact that once the electron has been excited, they will produce or they will release heat and also light fluorescence. That's why for plant scientists, especially the plant physiologists, if they want to check or if they want to measure at what level of photosynthesis takes place in the plant, they will check the level of heat as well as the light fluorescence reaction on the leaf. From the measurement, they may know the more the heat, the more the light fluorescence produced. It shows that the process of energy excitement in the photosynthesis takes place efficiently and effectively. Next, the photosystem 2 pull for replacement of electron from water molecule, which break apart into oxygen and hydrogen ion. The oxygen will leave the cell as in the form of oxygen, and hydrogen ion will contribute to the hydrogen ion gradient that drive the ATP synthesis later. For every 2 molecules of water that are split, 1 molecule of oxygen gas is produced. So now we know once the electron ejected from photosystem 2, the replacement of the ejected electron will come in from the water molecule, which break apart into oxygen and hydrogen ion. And this oxygen will leave the cell as in the form of oxygen gas. Meanwhile, the hydrogen ion will contribute to the hydrogen gradient that drive the ATP synthesis later. And for every 2 molecules of water that are split, one molecule of oxygen gas will produce. And now, the electron which was ejected from photosystem 2, they will enter electron transport chain 2, which is also in the thylakoid membrane. Every electron, once ejected from photosystem 2 by light, they will come out of the photosystem 2 and hopping from one molecule pigment into another molecule pigment, which is in the electron transport chain 2 in the thylakoid membrane. As I mentioned before, energy was lost by the electron as they move through the electron transport chain 2, which causes hydrogen ion to be pumped from the stroma into the thylakoid compartment. Hydrogen ion gradient form across the thylakoid membrane. This will be used to generate ATP later. I repeat again, energy was lost by the electron as this electron moved through the electron transport chain 2, which causes hydrogen ion to be pumped from the stroma into the thylakoid compartment. The pump or the channel of hydrogen ion from stroma into the thylakoid compartment will create a gradient across the thylakoid membrane. And this gradient across the thylakoid membrane will facilitate of the generation of ATP later. Meanwhile, light has also will be striking the pigment molecules in photosystem 1 as it happens to photosystem 2. It means that light energy will also eject electron from photosystem 1. The replacement of this electron will come from electron transport chain 2. So now we know the electron which was initially ejected from photosystem 2 will hop from one molecule to another molecule pigment within the electron transport chain 2 until they reach to the reaction center, followed by photosystem 1 will also ejecting the electron into the electron transport chain 1. The replacement of ejected energy or electron in photosystem 2 will come from the water molecule. Meanwhile, the replacement of electron ejected from photosystem 1 will come from electron transport chain 2. It shows that photosystem 2 and photosystem 1 are related in the form of electron supply chain. And now the electron will move through electron transport chain 1 once this electron ejected from photosystem 1. This electron, they will combine with NADPH and hydrogen ion to form NADPH, which is energy carrier molecule to be formed. They will pick up two energetic electrons along with one hydrogen ion. I repeat again, the electron will now move through electron transport chain 1 after ejected from photosystem 1. This electron, they will combine with an ADP and hydrogen ion, and the NADPH will form. Along the line, they will pick up two energetic electrons together with one hydrogen ion. And finally, this hydrogen ion in the thylakoid compartment will be propelled through the interior of ATP synthesis by the gradient across the thylakoid membrane. Hydrogen ion flow will cause ATP synthesis to attach the phosphate to ADP so that the ATP will form in the stroma. So this is the product of like reaction process. By having like energy, by having H2O or water going through photosystem 2, electron transport chain 2, photosystem 1 and electron transport chain 1, they will produce NADPH as well as ATP. This will be used in the Calvin cycle in order to produce sucrose. And now we move on to Calvin cycle. After we know what was happened in like reaction process, and now we move on to Calvin cycle. In this process, they convert all of the supplied chemical energy in the form of ATP as well as NADPH to be stored in sugar molecule. In Calvin cycle, first of all, they need CO2. Therefore, the process will capture carbon dioxide. The ATP and NADPH synthesized during the like reaction will dissolve in the stroma. These energy carriers will power the synthesis of simple 3-carbon sugar called glyceraldehyde 3-phosphate from CO2. The cycle is catalyzed by enzyme dissolved in stroma. For every 3 carbon dioxide molecule captured by Calvin cycle, 1 molecule of glyceraldehyde 3-phosphate or G3P is produced. So now we know Calvin cycle is a process where converting chemical energy in the form of ATP and NADPH, which was initially produced by like reaction to be stored in sugar molecule. And this process requires carbon dioxide. Why Calvin cycle to be known as cycle? there must be some things to do with the origin and the product, and the process will take place over and over again in a chain reaction and in a cycle process. Now, Calvin cycle is known as Calvin cycle because it is a cycle where it begins and ends with the same molecule of 5-carbon sugar known as ribulose biphosphate. Ribulose biphosphate is recycled continuously. They begin with having ribulose biphosphate and they end the process by having ribulose biphosphate. And Calvin cycle is best to be understood if we divide it into 3 different parts or phases, namely fixation phase, reduction phase, as well as regeneration phase. We will go through these 3 phases in detail in the next few slides. This is the overall features of Calvin cycle involving 3 distinguished phases known as fixation, reduction, and regeneration. In the first phase of fixation, it is a process where fixing carbon dioxide. In the second phase of reduction, it is to reduce NADPH becoming NADP plus hydrogen to produce glyceraldehyde 3-phosphate. And in the third phase of regeneration is to regenerate ribulose biphosphate from glyceraldehyde 3-phosphate. And the process will take place in cycle where they begin with having ribulose biphosphate and they end the process by producing ribulose biphosphate. And in between of these 3 phases, they are distinguished process involving reduction, oxidation, production, of different types of chemical molecule. In the first phase of Calvin cycle, which is a fixation phase, it is a process of phase to fix carbon dioxide. The cycle begins when carbon dioxide reacts with ribulose biphosphate. This phase will fix carbon and produce molecule of 3-phosphoglycerates. The cycle uses the enzyme Rubisco to combine the carbon dioxide molecule with ribulose biphosphate molecule. I repeat again, in fixation phase, the phase begins when carbon dioxide reacts with ribulose biphosphate. And this phase will fix carbon and produce molecule of 3-phosphoglycerate. The process will use enzyme Rubisco to combine the carbon dioxide with ribulose biphosphate. In a reduction phase, it is a phase where a reduction of 3-phosphoglycerate, which is the product of fixation phase, into glyceraldehyde 3-phosphate. 3-phosphoglycerate is phosphorylated by ATP followed by reduction of electron from NADPH. The product is the phosphorylated sugar, namely glyceraldehyde 3-phosphate or G3P. Some of the G3P or glyceraldehyde 3-phosphate synthesized will be drawn off to manufacture glucose and fructose. I repeat again, in reduction phase, it is a phase where the reduction of 3-phosphoglycerate, a product of fixation, into glyceraldehyde 3-phosphate. And this 3-phosphoglycerate, which was initially a product of fixation, will be phosphorylated by ATP and reduced by electron from NADPH. The product is the phosphorylated sugar in the form of glyceraldehyde 3-phosphate. And some of this glyceraldehyde 3-phosphate, which was synthesized in the reduction phase, will be drawn off to manufacture or to produce other glucose and fructose. Whereas in the final phase, which is a regeneration phase, it is a phase where to regenerate a ribulose biphosphate from glyceraldehyde 3-phosphate. In this phase, 5 out of 6 glyceraldehyde 3-phosphate molecules, which were produced by the reduction phase, used to regenerate ribulose biphosphate. This is because it is necessary and a must in order for the cycle to take place again. Meanwhile, the remaining one molecule of glyceraldehyde 3-phosphate, which is the product of photosynthesis, will exit the cycle. And now we know in the final phase, the product of photosynthesis will exit the cycle and produce to be used in the form of glucose. Normally, the remaining one molecule of glyceraldehyde 3-phosphate, which was exit the cycle, they may be used to form sucrose, a disaccharide storage molecule consisting of glucose which link to form a fructose. Or they may link together in long chain to form starch. This may be another storage form of molecule. Or they will form a cellulose, a major component of plant cell wall. Most of the glucose synthesis from glyceraldehyde 3-phosphate and the subsequent complex molecule synthesis from glucose occurs outside the chloroplast. Therefore, we may conclude at the end of the photosynthesis process, not only sucrose are being formed, but they may become a starch or even a cellulose. But the main formation of photosynthesis product is the sucrose. And overall, this is the Calvin cycle summary involving phases of fixation, reduction, and regeneration. During the fixation phase, 3 molecules of ribulose biphosphate combining with 3 molecules of carbon dioxide will produce 6 molecules of 3-phosphoglycerate. And during the reduction phase, these 6 molecules of 3-phosphoglycerate will be reduced by 6 molecules of ATP together with 6 molecules of NADPH to form 6 molecules of glyceraldehyde 3-phosphate. And out of these 6 molecules of glyceraldehyde 3-phosphate, 5 molecules of glyceraldehyde 3-phosphate will proceed into the regeneration phase. Meanwhile, the remaining of 1 molecule of glyceraldehyde 3-phosphate will exit the cycle to form glucose. And in the last or final phase in Calvin cycle, which is regeneration phase, 5 molecule glyceraldehyde 3-phosphate will combine with 3 molecule of ATP. They will produce 3 molecule of ribulose biphosphate to be used in fixation. Therefore, the cycle complete and it will take place over and over again as long as the 3-phosphoglycerate can be produced through fixation, as long as 6 molecules of glyceraldehyde 3-phosphate can be produced through reduction, and as well as ribulose biphosphate can be produced through regeneration.